This unit is going to be about the way cells divide. Cell division, also called cell reproduction, is when a parent cell it splits into two other cells. We call the parent cell, the original cell, the parent cell. We call the two new cells after division daughter cells. Okay, we talked about how bacteria and archaea do cell division. It's called binary fission. Again, that's asexual reproduction, where the daughter cells are identical to the parent cell, clones of each other. Eukaryotes can do either asexual or sexual reproduction. They can do something similar to binary fission, that's called mitosis, where the mother cell forms two identical daughter cells, identical to the parent cell. Now, the cells that do this are called somatic cells. The cells they become are somatic cells as well. This is a form of asexual reproduction. Our cells multiply. Our body cells or our somatic cells multiply by mitosis. This is asexual reproduction, just like binary fission, except there's more than one chromosome involved. In mitosis, the daughter cells have the same number of chromosomes as the parent cell had. Okay, in meiosis is a process that forms gametes or sex cells. Gametes can be sperm or egg. Sperm, of course, is the male version of the gamete, and eggs are the female version. These are the symbols for male and female. It's important to note that the process of meiosis, the formation of the gametes, the daughter cells have half the amount of chromosomes that the mother cell has. So you have a mother cell with, let's say it has two chromosomes. Well, actually, it has two pairs of chromosomes. That's called a diploid situation. And what happens is there's a first division with each cell getting half the number of chromosomes. And then there's a second division where the daughter cells get the same number of chromosomes, but half those chromosomes that were in the first division. We'll talk more about this later in this unit. So you get so here we have two pairs of chromosomes. Actually, 4 chromosomes in the mother cell. And then the daughter cells, after the first division, you got 2 chromosomes in each daughter cell. And in the second meiotic division, you get 2 chromosomes still, but only half per daughter cell. So the result is you get 4 daughter cells, each with half the number of chromosomes that the mother cell had, but 1/4 or 1/4 of the total DNA that the mother cell had. Okay, so it's very important to know the difference between diploid and haploid. Diploid is symbolized as 2 lowercase n. Haploid is symbolized by a lowercase n. Okay, haploid means you have 1 copy of each chromosome that there are in the parent cell. Diploid means you have 2 copies of each chromosome, like the parent cell had. Okay, so one of these things is representing that could have 2 parts like this. So one of those is representing actually 2 copies of DNA. So one of these guys right here is what I represented as an X. Now, if you have 2 of them, they have the same genes. For example, hair color, eye color, hair texture, height. Those traits are all on both chromosomes in the pair. Well, there could be different varieties of those genes. For example, this gene might have blue eye variation for eye color, and this gene might have brown eye variation for eye color, but the eye color gene is on both chromosomes, on that pair only. Okay, we call a pair of chromosomes a pair of homologous chromosomes. So these are not homologous chromosomes. These are a homologous pair of chromosomes. So if you have the gene for hair color right here, say you red hair, this form of that red hair gene on the same chromosome has to be red hair. It can't be brown hair, at least before meiosis occurs. Now, you could have this, you have to have the same gene over here, but it could be a different form. So let's say on this chromosome in the homologous pair, instead of red hair, you had black hair gene. Well, this would have to be a black hair gene as well. So the gene is hair color. It's a trait for hair color, let's say. But there's variations of that gene, which we'll call alleles later. But you could have different varieties of a gene on the 2 chromosomes in a homologous pair. But in a single chromosome, you would only have one variety of that gene. For example, you can have red hair gene and black hair gene on the same chromosome. But the other chromosome in the homologous pair could have, for that same gene, have a different variety. For example, black hair. So, you can't have a combination of red and black forms of, for example, the hair gene on the same chromosome. So if you're going to have black hair genes on a chromosome, then you have to have both genes on that chromosome in that homologous pair being black. You can't have a combination of red and black forms of the hair color gene or any gene on the same chromosome. But on two chromosomes in a homologous pair, you can have different varieties of the same gene, like I have shown here. Okay, just to remind you, diploid means you have a pair, you have homologous pairs. The cell has homologous pairs if it's diploid of chromosomes. So right here, these guys, you see pairs of homologous chromosomes. So that's a diploid situation. Right here, you see one of each type of chromosome. You don't see any pairs. So if you have no homologous pairs of chromosomes, you're haploid. So what happens in meiosis is diploid forms two haploids, and then those two haploid cells split up to form haploid cells. So once again, here's the haploid mother cell. I'm sorry, the diploid mother cell. Let's just use two chromosomes. Squiggly little one. Okay, so that's 2N. Then what happens, you break it up. The first meiotic, this is meiosis. The first meiotic division is you get the pairs separate. So, but you still have two chromosomes like the parent cell had, but you don't have homologous pairs. These aren't pairs in the first division, so they're haploid. And then the second meiotic division is when the chromosome splits. Like I showed before. Now you still have two chromosomes, but you don't have, those chromosomes don't have they're not X's, they're single arms of chromosomes, so they're like one of these, not both of them together. Okay, so these are also haploid. Now, in mitosis, That was meiosis. In mitosis, it's much simpler. You have your you got your pairs of chromosomes. And what happens is it breaks up into daughter cells that still have the pairs. Still have the pairs, but they're only one branch of the chromosome, one half of the chromosome that was there. So this is still 2N. We got 4. We still have 4 chromosomes, but they're only one branch. Okay, so they're still diploid. You still have pairs. You have 2 pairs of chromosomes. So that's mitosis. There's a big difference. Now, before we go on, if this is a chromosome, okay, this is a homologous pair of chromosomes. Okay, they have the same genes in them, but they could have different varieties between them of that gene. Now, when we look at, this is only one chromosome. This picture I just showed you had 2 chromosomes. And it would also be considered diploid because I have a pair of chromosomes. So here we see 2 chromosomes because they're the same genes in both chromosomes. That's a homologous pair of chromosomes. Now right here, just one of those guys is 1 chromosome. Now I've been calling these guys arms. Now in a chromosome, the 2 arms are identical. The genes are the same. The forms of the genes, the varieties of the gene, the variety is the same on both sides. So if you had red hair color here, you have a red hair color gene there, like I said before. But this is called, each of these things right here, no matter what kind of chromosome you have, each of these arms is called a chromatid. So here's another chromatid here. This whole thing. So we have 2 chromatids. So this is a 2 chromatid chromosome. Right here we have 2 chromosomes. We have 1, 2, 3, 4 chromatids. Right here, this is 1 chromosome. Don't call it half a chromosome. But that chromosome only has 1 chromatid. Okay, he has all the genes that a 2 chromatid chromosome has. So, but he only has half the amount of DNA. So, a 2 chromatid chromosome has copies of each gene on 1 chromatid that are on the other chromatid. But a 1 chromatid chromosome only has 1 copy of each gene. Okay, so let's get a little more general. Let's go back to mitosis. In mitosis, the parent cell forms 2 daughter cells. And parent cell is diploid. The daughter cells are diploid. That means what? They have pairs of each chromosome. After mitosis has occurred, the daughter cells are identical to each other and identical to the parent cell. Both parent cells and daughter cells are somatic, or body cells. Mitosis occurs everywhere in our body. Skin cells, muscle cells, anywhere a cell needs to be replaced. When you're growing, you're making new cells. Think of a little baby turning into a toddler, turning into a teenager. That's a lot of new cells forming, and that happens by mitosis from old cells. Meiosis only occurs in our sex organs. So the testes, if you're a male, the ovaries, if you're a female. These are the sex organs. Okay, so there we have the parent cell is called a germ cell for some reason. I don't know. It's not a germ, but the parent cell is diploid, or the germ cell in the sex organ is diploid. Now, what happens is it undergoes two divisions. So, like I showed you, forming 4 unique daughter cells. They have the same genes, but not necessarily the same variety of genes. And these cells are haploid. So you went from diploid to haploid daughter cells. These cells are called gametes, or sex cells. Okay, so cell division. These are reasons that we use mitosis. Now, we can't reproduce asexually. We can't. We only can reproduce sexually, but simpler organisms can. Like bacteria for sure, that's all they can do. Okay, we reproduce sexually, and that requires meiosis to form the gametes, or the sex cells. You can't have sex without gametes. Again, mitosis is diploid forming diploid. Meiosis is diploid forming haploid cells. Okay, this thing is called the cell cycle. And right here in gold is mitosis. There's phases of mitosis. We'll talk about those, starting with prophase, metaphase, anaphase, telophase, and then the cell cytoplasm and organelles are divided up between the two daughter cells. That's called cytokinesis. Okay, so mitosis, let's just look at that without getting into too much detail. Mitosis is this part. In mitosis, the nucleus divides, splits in half. But more specifically, the nucleus divides, yeah. But more specifically, the each chromosome, each 2 chromatid chromosome is split. So what happens is each chromosome, let's say you have a chromosome pair like this. What happens is each of those guys is broken off. And if that's the parent cell, pair of chromosomes, this gets divided. Each of these gets divided. I should make these different colors. So that the daughter cells will get pairs, but only one chromatid from each chromosome in the pair. So if this was the 2N cell, what happens during mitosis forms two daughter cells. They're still 2N because they have homologous pairs, but they only have half the DNA that the mother cell had before mitosis. So that's the purpose of mitosis, is to divide the nucleus equally. Now, later on, during interphase, that DNA will replicate and go back to diploid cells. So I just wrote this. Each daughter cell gets one chromatid chromosome for each homologous pair of the mother cell. So for each homologous pair, you get half the chromosome, but these two halves are identical, right? The two halves of the black chromosome are identical genes, and identical form of the genes. Same with the blue chromosome. Okay, the cytokinesis stage of cell division is when the cell, the cytoplasm, and the organelles roughly subdivide. So here's your cell with, let's say, with your two new nuclei. I'm just drawing two pairs of homologous chromosomes, and what's going to happen is that's going to subdivide. The cytoplasm is going to, with the organelles, is going to subdivide. And there's your nucleuses, your two nucleuses, and form two new cells. That's called cytokinesis. So the cytoplasm and organelles are roughly split up between the two cells. Roughly, that doesn't have to be precise. The two new cells, two new daughter cells. So both the nuclear division called mitosis and cytokinesis are part of what is called cell division. The time a cell spends between daughter cell and getting ready to do cell division to form two new daughter cells, that time period is called the interphase. Is between the cell division phases. Okay, so what has to happen, the most important thing that has to happen, is each chromosome, the DNA is doubled, and it goes from 2N to 2N. Yeah, but with DNA copying itself or replicating, when the DNA doubles or replicates, you don't get more chromosomes. Rather, you get more chromatids. So, like one cell here has 2 chromosomes, and then this cell still has 2 chromosomes, but instead of 2 chromatids, now it has each chromosome that was there before now has another chromatid. Okay, you can see that interphase is broken up into 3 stages. S phase is DNA synthesis. That's what the S stands for. And the G stand for the gap in between DNA synthesis or replication. Okay, so there's a gap 1, there's the S phase, and then there's gap 2 of interphase. So cells spend more than 90% of their life in interphase. So cell division takes a short time. So in this cell cycle, you can see this would be the cell division phase. They're not that includes mitosis and cytokinesis. They should add it. Now some cells will leave the cycle and never undergo mitosis cell division again. This is called the GO phase. And cells like nerve cells would do that. Nerve cells don't, once they're formed, they don't do mitosis. They grow, but they don't do mitosis anymore. So if you damage your nerve cells, it's kind of irreparable. You can't replace them. Okay, so certain cells do that. Muscle cells also. Muscle cells are like that too. But most cells, after the mitotic phase, they go into G1. That's what we're going to talk about, gap 1 phase. I like to think G is growth because that's what's happening. So the cell increases in volume. It keeps on making more cytoplasm and organelles. Okay, it makes proteins necessary for DNA synthesis, replication, and like I said, makes more organelles. So we're now in S phase. S phase is DNA synthesis. DNA replication occurs. That's where the 1 chromatid chromosomes become 2 chromatid chromosomes because the DNA has replicated. So each of those 1 chromatid chromosomes now becomes 2 chromatid chromosomes. Now you don't see the chromosomes because they're in a condensed form. I'm sorry, they're in an uncondensed form or a soupy form. A lot of the histone proteins have come off the DNA molecule, so it unwinds. You don't see it under the microscope. So here's a picture showing what happens to the chromosomes if you could see them during the S phase. Okay, centromere is the thing that connects the two chromatids, if there are two chromatids. Okay, so it's that little black dot, like the belt, that holds the two chromosomes, the chromatids together. Okay, so here it is before the S phase. Before the S phase, we have two chromosomes, homologous chromosomes, but only one chromatid each. Then during the S phase, what happens is that each chromatid replicates itself. Well, actually, the DNA replicates itself. And so we get each chromosome now having a sister chromatid. Don't get sister chromosomes and sister chromatids mixed up. I don't really use sister chromosomes. I use homologous chromosomes. Then I use sister chromatids, the word sister. Then G2 phase, it keeps on making more organelles. It makes the proteins that are needed for the mitotic phase. Then the phases of mitosis will occur. PMAT, prophase, metaphase, anaphase, telophase. And then cytokinesis will follow. So meiosis will occur, then cytokinesis, they should have put that in there, will divide the cytoplasm organelles into two portions, two new cells. The stages of mitosis are prophase, metaphase, anaphase, telophase. Now, you can have something called prometaphase. That means before metaphase, but I kind of lump these together. Okay, so let's talk about each phase now. Okay, so here's the cell in the middle, right here. This thing right here. Okay, so we see organelles. We see the cell nucleus right here. Cell nucleus right here. Here's some endoplasmic reticulum. Here's some mitochondria. Here's some vesicles coming off the ER. These things I kind of avoided. These are the centrioles or the centrosome. The area is the centrosome. The bodies, if you see them, those little perpendicular orange things, those are the centrioles. What these guys do is spin. They shoot off microtubules, and those will connect to the chromosomes eventually and organize them and get them ready for separation. Okay, we'll talk about that in a little bit. So what happens is the cell membrane starts to starts to dissolve, starts to break off. The nucleolus, where all these little speckles here, it used to be a body, a location. It spreads out, it disappears. The soupy chromosome, the soupy DNA, condenses into chromosomes. You see chromosomes starting to form, whereas you couldn't see them before. And what you see, because it's after synthesis, you see homologous chromosomes with each chromosome having two sister chromatids. Okay, the centrosomes, or the centrioles, move to opposite sides of the cell. First they divide. There's only one pair usually. Then they divide, and then they'll move to opposite poles of the cell. And you can see the centrosomes starting to form these microtubules. Okay, that microtubule will form a web that captures the chromosomes and moves them, called the spindle apparatus. I call those spindle fibers. So the region around a chromosome near the centromere is called a kinetic core. That's where the spindle fibers will connect. So you see this guy right here. You got spindle fibers starting to connect or having connected to the kinetic core. Okay, so if you see a chromosome like this, here's the centromere, and here's the kinetic core. And it's just a place where the microtubules can attach. Once the microtubules have attached, these chromosomes will be pushed or pulled to the middle of the cell, like the equator of the cell. Now, they call this prometaphase because it looks like you don't see the cell membrane at all. All the chromosomes are distinct and they're close to the middle of the cell. It's right before metaphase, but I still call it prophase. Okay, here you see a actual picture of a cell with the different kinds of proteins stained with different colors of dyes, fluorescent dyes. The spindle fibers are dyed green. The chromosomes are dyed blue. The histone proteins on the chromosomes are dyed blue. And the red things are the pink things are the kinetic cores, where the spindle fibers are connecting to. Okay, so the centromeres, or the centrosome, would be where all the green fibers are coming out. So we can see that the centromeres are anchored to the cytoskeleton, or even the cell membrane. The cell membrane, not the nuclear membrane. So right here, they're anchored and they're pulling at the chromosomes. They're pulling or pushing at chromosomes, so they're all in the middle equator of the cell. So here's another picture showing the kinetochore is the region where the spindle fibers connect. And the central mirror would be that little belt that I showed. We got the sister chromatids, and this is one chromosome, not two. Okay, so at metaphase, the spindle is complete. Each chromosome is connected on both sides of the kinetochore by both centrioles or centrosomes. They're right in the middle of the cell, the midplate. They call that the equator. They call it the metaphase plate. Meta means middle. Okay, those chromosomes are going to be mixing. They're going to be pulled apart. Those chromatids are going to be pulled apart. But that's in the next stage called anaphase. Okay, so these spindle fibers are pulling the X's, and the central mirror splits as they're tugged apart on both ends of the kinetochore, and the chromosomes move towards the opposite sides of the cell. So the sister chromatids separate at the central mirror and are pulled apart. Ana means apart. Anaphase. So in animal cells, these protein, there's a ring that forms that's causing this furrowing effect. And it's called the cleavage furrow, this separation between the cells. And it's caused because these fibers, this ring of fibers that can contract, much like muscle fibers, they keep on making this ring tighter and tighter and tighter as they contract until the two cells are torn apart from each other. This is called a contractile ring, and that happens during cytokinesis in animal cells. So in plant cells, you can't use a contractile ring because there's a solid cellulose wall around the membrane. So what happens is that vesicles with cellulose in them are formed, and they fuse with each other in the midplate of the cell where the mitotic plate was, and those fuse together and eventually form a new cell wall separating the cells, and the cells continue to grow like this as all this is happening with new material being added to the cell wall. Okay, so here's just a summary of the different stages. Notice that they call this the mitotic phase. It includes mitosis and cytokinesis. I like to call it the cell division phase. Okay, this is kind of a general question. What is not a function of the cell cycle? Okay, cell cycle is mitosis and interphase. So growth occurs during interphase, so that's not an answer. That is a function of the cell cycle. Generation of lost parts. Well, if you lose a mitochondria because it gets damaged somehow, yeah, that's during interphase that happens. Or if you need more cells, like you got a cut on your hand and you lost cells that way, new cells have to be produced from other cells that are still there. So that's mitosis or interphase of his organelle. So that's not an answer. That is a function of the cell cycle. Tissue repair, same thing. So all these and more are functions of the cell cycle. The only thing I would think of that wouldn't be would be the formation of sex cells. I don't see that as a choice. Which occurs during interphase, division of the cell. No, division of nucleus, yeah, but not of the cell. Cell growth and duplication of the chromosomes, replication of the DNA, yes. Reduction in the size of the cell, why would it reduce? Why would you reduce organelles if you're going to make 2 new cells? So how do eukaryotic chromosomes differ from prokaryotic chromosomes? Well, the chromosomes are housed in the nucleus in a eukaryotic cell, but in a prokaryotic cell, they aren't. So they're looking for what features do eukaryotic chromosomes have that prokaryotes don't. Eukaryotes aren't circular. Eukaryotic chromosomes aren't simpler. And yes, they're copied during cell division, but so are prokaryote chromosomes. Remember, 90% of the time or more that a cell spends in a cell cycle is in interphase. If I ask you what is the shortest phase, the one that takes the least time, the answer would be anaphase. Because that's where the chromosomes split and quickly get pulled to the ends of the cell, 2 opposite ends. Okay, if the synthesis phase, that means where the DNA is copied, daughter cells would have half the genetic material found in the parental cell. So here is a nucleus right before the S phase, or right after mitosis. That's one of the daughter cell nuclei. Okay, so what should happen during the S phase is each of these guys get a sister chromatid. No, that did not happen. When mitosis occurs again, okay, if that happens, when mitosis occurs again, we get this guy right here. No, that did not happen. If this did not happen, just stay like this, it's only half the DNA that it should have. Okay, the nuclear membrane starts to break up in prophase. This is necessary so that the spindle fibers can connect to the chromosomes and pull them to the center of the cell, the metaphase plate. Okay, the kinetochore makes the spindle? No. The kinetic cord attaches the spindle fibers. Kinetic cord attaches spindle. Centrosome holds the chromatids together. No. The centromere does that. Okay, the centrosome makes the spindle. Okay, remember those little guys like this, the centrioles. And the centrosome is the area between around them, and it makes the spindles. And the centromere is the belt that holds the chromatids together in a chromosome. So that's the correct choice. Chromosomes line up in the middle, the mitotic plane. That is called middle, metaphase. Okay, nuclear membrane forms around the new nucleus. At the end of mitosis, that would be telophase. Process by which the cytoplasm is subdivided between the two new cells is cytokinesis. So there are checkpoints throughout the cell cycle. It's like a quality control system that detects either mistakes during replication or a mistake in how the chromosomes are being attached to the spindle and moved to the metaphase plate. If there's a mistake that's sensed, the cell tries to repair it, and then it keeps on going through the cycle. But if the cell cannot repair it, the cell self-destructs. A death program is initiated, killing the cell. So he doesn't keep on reproducing. Normally, when cells come in contact with other cells after mitosis, they go into GO because you don't want cells to keep on dividing after like the space is filled where they should be. So let's say you get a cut. So the cells sense that there is a need to divide and fill that space made by the cut. And so they'll come out of GO, they'll be signaled to come out of the GO stage, and they'll divide. Nerve cells don't do this, or muscle cells, but let's think about skin cells. Okay, so once they divide and come into contact with other cells as that space is filled, they'll go back into GO. Now, cancer cells are normal cells, except that when they touch each other, they refuse to go into GO, and they keep on dividing. And that's not good because you can get a tumor, which is the mass of these cells that keep on growing when they're supposed to not be growing. And this tumor can take blood away from other cells that need to be there and actually push into those cells and cause issues. So I think some tumors are benign in the sense that a surgeon can cut them out without hurting the surrounding cells. Some are malignant in the sense that they're growing like at a place that if a surgeon tried to remove it, it would hurt other cells that don't come back, like nerve cells. So if you had a tumor in your brain, it's malignant in the sense that if it continues to grow, it will push out normal nerve cells, and you'll lose your brain functions. Okay, also tumor cells can be, malignant tumors will get to a point where they metastasize. That means they shed off cells. They send off little cells into the bloodstream that travel to other parts of the body and then form tumors there. It's called metastasis, and that's really hard to control once that goes. So this property of cells, normal property of checkpointing, when you touch another thing, another cell, and go into GO, is called contact inhibition. And cancer cells somehow ignore contact and continue to divide anyway. So here we're talking about malignant versus benign tumors. Benign tumors are not life-threatening. Malignant tumors either are causing damage to surrounding tissues or sending out cells, cancer cells, in the bloodstream that will find other places to grow into malignant tumors. In other words, metastasizing. So you could theoretically get a little bit of skin cancer, and if it's not removed, it could do metastasis and send off cancer cells through the bloodstream that will grow in your lymph nodes, where blood is filtered, can form in your lungs, it can form in your brain, your liver, your pancreas, inside your bones. So it's important that you do see, especially in Florida, you see a dermatologist to find any skin cancer before it gets to the metastasis stage. Okay, so this one, nerve cells are in permanent GO stage. So there are checkpoints at different places in interphase, whether it's gap 1 or gap 2, and during mitosis actually as well. So before mitosis, that'd be gap 2. So all the above are correct. Okay, a benign tumor differs from malignant tumor. Well, benign tumor is cancer, but a benign tumor is not metastasizing. Metastasizing would be spreading from its original place, and it can cause health problems. I mean, it needs to be looked at. It needs to be monitored so it doesn't turn malignant. So I shouldn't put letter D either. Which of the following types cells spend less than 9% of its time in interphase. Well, looking at the choices, a nerve cell spends much more time than 90% because it's permanently in interphase. Muscle cell, same thing. Cancer cell, it's always dividing, so it spends very little time in interphase. Yeah, contact inhibition. If you ignore contact inhibition, a cell will continue to divide even when it's touching other cells, forming a tumor. So this is true